Any other hormone research proposals?
Let me make a clarification here, because this is one of the things that I see in the literature that is just boundless. When we talk about supplements or we talk about any type of hormonal therapies, what's sorely lacking is basic pharmacology, dose-response curves. When you get into vitamin therapy, again, you talk about, is it 60 milligrams of vitamin C? We found it wasn't effective. Was it 600 milligrams? Was it 6 grams? Was it 60 grams? And then you get all these things. You cannot compare these. You can't do any retrospective studies. You can't do any meta studies on this. So if you want to do this correctly, first of all, you need to look at real good dose-response curves. That's expensive. Then if you look at vitamin therapy, I think you're probably going to get into something like combinatorial math, because it's been shown by computer modelizations that if you look at, say, drug interactions, you can model drug-drug interactions. You might be able to model drug-drug-drug interactions, but once you get to 4 drugs, the combinatorial math is just too enormous, and you cannot predict the side effects beyond 4 drugs. So this becomes a problem of, first, basic pharmacology, which is expensive, dose-response curves, and number 2 is the combinatorial math of how you're going to combine these. And that's what's going to limit, the effectiveness of it. Because if you have a study, and let me just give a silly example, because I give this example all the time. If you do if you give, say, 1 milligram of Lipitor, and nothing happens, the headline is, Lipitor shown to be an ineffective drug. If you give people 100 milligrams of Lipitor, the headline will be, Lipitor found to be a terribly destructive drug. If you do dose response, what happens at 5 milligrams, 10 milligrams, 15 milligrams, 20 milligrams, and you look at the markers, which they're not doing, because all they're looking at is LDL, and they're not looking at the other inflammatory markers. So it shows the folly of not paying attention to basic pharmacology, and then again, the combinatorial math.
I'll just point out to our note taker, is that you, Kevin? That in fact, those issues are more general than hormones. Those are the supplements and all through the optimization of metabolism. So I'd like it.
And I'd just like to add one thing to that proposal, which I think is a very important one, which is that what you say is particularly true for things that are off patent, things that are natural products, for example, things that drug companies are not going to make money out of getting any more knowledge about, because those things, even one dose tends not to be tested properly, especially with markers, as you were saying. So I think with particular, I absolutely just want to endorse what you said, but with particular emphasis for things that are not commercially promising.
Excellent. Any other comments or suggestions under the heading of optimizing metabolism? Okay, given that there aren't any, we will now move on to repairing damage. I have a whole bunch of things under this heading, but let's start with telomeres.
Slide number 10.
Very good. Let's see, do we have, yes. Shall we do it in this order?
It doesn't matter.
No, I mean, it would be convenient to do it that way. That way we all know what we're talking about. Item number 1, pill to fix extracellular glycation and cross-linking. John, I'll let you kick it off.
So I've been using the word pill kind of loosely and colloquially, but in fact, pill could mean a combination of things, because there's several types of glycation and cross-linking. There's one chemical that's already been semi-proven to be effective against one kind of cross-linking. There's another kind of cross-linking that really needs to be tested for high-throughput screening to develop. But I think that could be done within about 2 years for about $5-6 million, and then at that point it would be a matter of clinical trials to get it through FDA and so forth. That's another 5-6 years and another $50 million. So that's my take on extracellular glycation and cross-linking in terms of chemical fixes. But it leads right into point number 2, which is improving extracellular matrix turnover by stimulating lysosomal or other cellular action. And that's what I talked about yesterday, so I won't go too much more into that now.
Thank you for your concision. Topic number 3, amyloid. Who will take amyloid?
There's, of course, a well-known type of accumulating junk in the brain called senile plaques in Alzheimer's disease, but this is just one example of quite a range of different types of garbage that accumulate in the extracellular space in various different tissues. Things that accumulate in different tissues tend to have a different molecular structure. They tend to be composed of different things, but their superstructure, the sort of tertiary structure, quaternary structure that they tend to adopt, has a lot in common. That's why they go under this general heading of amyloid. And the approach that I think has the most promise at the moment in addressing the problem of amyloid in the brain is immunotherapy against it, which Steve Cole's talked about a little bit, and I guess Steve's going to want to say a few words after I've finished it in a moment. But as far as I know, there is still very, very little immunotherapy going on, immunotherapy research going on against other amyloids. Steve spoke about one or two interesting things, which I want him to perhaps go on to in terms of more specific projects. But what I'm not seeing is any attempt to really emulate the approach that has now already got to phase 3 for amyloid in the brain, again, in Alzheimer's disease, namely passive vaccination, the creation of antibodies against amyloid and injection of antibodies into the tissue that's affected so as to activate the immune system against it. I think we could easily, with as little as a couple of $1 million, kickstart some extremely promising projects against amyloid in the pancreas, for example, which is a very major player in the later stages of type 2 diabetes, and also against the amyloid that Steve spoke about yesterday that affects particularly the heart.
Oh, please, briefly.
In addition to that, there is a drug in clinical trials by an Australian company called Prana that seems to be effective in dissociating not only brain amyloids, but also perhaps pancreatic amyloids and some other. It hasn't been tested on TTR, but if to the extent that they aggregate by similar mechanisms, that drug or something in a similar class might be effective, and the company's name is Prana.
Thank you very much, Steve Coles.
Yeah, with respect to the problem of amyloidosis in the brain, we know that beta amyloid is the primary constituent, along with tau tangles, of the plaques that occur sort of randomly distributed, especially in the hippocampus, which is the part of the brain that has to do with consolidation of memory. And there was a man and wife team in Germany, both physicians, Brock and Brock, B-R-A-A-K, and they have this Roman number system for measuring the density of these lesions in the brain, which were highly correlated with clinical Alzheimer's disease, but not 100%, in the sense that there were anomalies in the 2x2 matrix, so to speak. People who had lots of lesions in autopsy may not have shown clinical Alzheimer's disease. And then there were people with what looked like Alzheimer's disease that did not have a Brock score of 6 or 5, which is the upper end of the scale. It goes from 0 to 6. So it was complicated, and I tried to get a better understanding of this, and I'm working with Dr. Fiala, who is in the Department of Medicine doing an Alzheimer's study, is published extensively in the Alzheimer's literature, and he says there are really 2 types of amyloid in the brain, both of which are beta amyloid, but some of these are soluble and some of them are insoluble. The insoluble ones may be appearing in the brain without doing any damage really to the neurons, and they're innocent bystanders. Those are the things that you see in the Brock score, but there could be soluble...
So just to make it brief, amyloid is not necessarily amyloid.
Yeah. Soluble forms, and they could be in the neurons, and that's what kills the neuron.
Excellent, thank you. Number 4, pill to fix epigenetic alterations. That's pretty impressive that it'd be one pill, John.
Tom, can you flip to slide number 38 just for a moment? So this is some work that I saw reported at an Ellison meeting a couple of years ago, and I think it's still early days, but it appears that some of the HDAC inhibitors which are being explored for cancer are actually acting on chromatin remodeling, and to the extent that certain types of epigenetic alterations have to do with the way the DNA is folded around the histones, it's being suggested by a few people now that there may actually be a whole other genetic code, a whole alphabet of very specific HDACs and HDAC inhibitors which open and close different sections of the DNA, allowing expression or repression of different genes. I think it's important to encourage further research in this area, and whether it turns out to be a pill or an injection or some kind of gene therapy, I don't know yet.